this is a little bit of a interesting spot in the syllabus. If I ask most of you at the end of the semester, many of you will tell me that today is where we started talking about clinical immunology. If you ask me about the syllabus, I will tell you that Wednesday is where we start talking about clinical immunology, because this one is sort of a little bit of both. We are talking about some basic immunology phenomena today, but I'm going to be using examples of clinical conditions when talking about them. And so it's going to sort of feel a little bit like we're doing both. It's going to be fully into clinical stuff by Wednesday. You have previously seen the word tolerance, especially when we talked about central tolerance. And central tolerance is a way that we get rid of self-reactive responses. We stop a self-reactive response. And I will just point out, because this came up on some people's exams, which made grading interesting, because I realized that some of you were using this one word wrong, and I wasn't sure exactly how I taught you about that word, so I wasn't sure I could care that you were using it wrong. But I think it was making some things a little confusing. So oftentimes when we think of autoimmunity, it's sort of like immune response is too much. You're making extra immune response. Immunodeficient means you're making not enough. Immunodeficient isn't a broad term of my immune system doesn't work. Immunodeficient means something is missing. Your immune system is lacking something. And so it seemed like a few of you were confused about the term immunodeficient. So immunodeficient means something is lacking, something is not working in your immune system. So if we go back to Goldilocks, immunodeficient is the too little, autoimmunity is the too much kind of problem. And autoimmunity is the result of some failure of tolerance. This can be due to some genetic factors. This can be due to some environmental issues, either of which can alter immune regulation and lead to autoimmunity. We've talked about the ways that central tolerance happens in both B cells and T cells already. So central tolerance is a tolerance mechanism that happens during development of our B cells or T cells. And so this is a process that you've seen before in terms of B cell central tolerance. We saw that B cells in the bone marrow that were self-reactive either could be deleted, could be receptor edited by making a new light chain, or could become anergized. And in all three of these cases, we would end up with no self-reactivity in the periphery, either because we turned our B cell into a non-self-reactive B cell, or because we energized that B cell. But we also talked about the fact that there were a couple of problems with this scheme that left us open, potentially, to autoimmunity. So when we talked about B cell central tolerance, there was one big flaw we saw with B cell development that made central tolerance imperfect. What was the potential problem for B cells? Yeah, Alina.
They didn't have all types of antigens in the bone marrow like other parts of the body.
Exactly. So B cells are limited in testing against antigens that are in the bone marrow, and only some antigens are in the bone marrow. So B cell central tolerance is pretty good, but it has this flaw that we're only testing against the subset of antigens that are present in the bone marrow. There is one other big issue with B cells. And so I guess the issue, just to be clear here, we have all these B cells. They did not test against some antigens in the bone marrow. And so it's possible that self-reactive B cells could still leave because they didn't test against that self-antigen that they are reactive against. And so there's a way that these B cells are sort of not even slipping through the cracks, but a way that we are getting some self-reactive B cells out in the periphery. And these are requiring us to have other tolerance mechanisms. There's one other part of B cell biology that we've discussed that also makes makes tolerance in B cells a big deal and makes us say, oh my gosh, there better be a good peripheral tolerance mechanism for B cells because this thing is a problem. Can you guys imagine what that other potential problem might be? I would normally not ask you to imagine it, but if I remember correctly, once I taught you this thing, somebody guessed it that day anyway, so might as well give you the chance. So when we talked about the germinal center reaction, remember that in the germinal center, our B cell is going to undergo this process called somatic hypermutation. It's going to heavily mutate the V, D, and J segments of the heavy chain and the V and J segments of the light chain with the goal of making stronger affinity for antigen, with the goal of affinity maturation. And so our B cell will divide, it will make multiple new sort of mutations in its B cell receptor. It's entirely possible that when those mutations arise, some of the B cells that were previously not self-reactive will become self-reactive B cells. And so it's also possible that the germinal center reaction can make B cells that were previously not self-reactive into self-reactive B cells as we acquire these mutations. And so for both of these reasons, it's clear that peripheral tolerance is going to be a big deal for B cells. We also talked about central tolerance in T cells and how we induce tolerance during T cell development. There were some pros for T cells, some things that were sort of nice for the T cells, and also some things that were sort of a problem for the T cells. So did T cells have the option to receptor edit in order to get around self-reactivity? No. T cells don't receptor edit, so they don't have that option. They don't have the option of coming out of the thymus anergic. But do they have the problem of being limited on their antigens? What's the story with the antigens in the thymus? Yeah, Molly.
Make sure that they see basically every antigen they can before they head into the periphery, so they don't have the same B cell.
Exactly. So air leads to the transcription of all of these tissue-restricted antigens, and so you can sort of imagine that in the thymus, every potential antigen is made to test against our T cells, and so we can negatively select sort of any T cell that is highly self-reactive because of air. So the T cells don't have one of the problems that B cells have, but the T cells have another big problem. So what was the other big problem of T cells? Yeah, Emilio.
You want them to be, at least to a certain extent, self-reactive.
Yeah, so in order for a T cell to leave the thymus, it has to have low affinity for self-antigen. So there has to be some sort of low binding to self-antigen in order for that T cell to actually be positively selected and leave the thymus. This allows us to make sure that all of our T cells bind to self-MHC. And so, as we said, every T cell is a little bit self-reactive when it comes out of the thymus. And when we talked about this during our T cell development lecture, I'm pretty sure you all looked at me with the idea that you were all going to die. I'm pretty sure you were terrified at that point. There are patients who have mutations such that they cannot do central tolerance correctly. When a patient has a mutation so that central tolerance gets messed up, they have a big problem. because they don't just have one autoimmune disease, they will have multi-organ autoimmune diseases. They'll have multiple autoimmune diseases at the same time. I've shown you one example of this previously. It was a disease called APSED, which is in the upper right-hand corner, or autoimmune polyendocrinopathy in candidiasis with ectodermal dystrophy, APSED. In APSED, patients are missing air. This is actually how air was described. Thus, the tissue antigens are not made in the thymus, and self-reactive cells are not negatively selected on those antigens in the thymus. As a result, our patients have autoimmunity, but what's interesting about their autoimmunity is that they tend to have multiple different autoimmune diseases at the same time. Each patient has a slightly different combination. So you can see that you might have a patient who has hypoparathyroidism and adrenal failure and diabetes or something like that. So these patients have a combination of autoimmune diseases. The issue is that it's just a question of which T cells happen to make it out of the thymus and not get negatively selected as they should have. These patients also have some weird things going on with their fingernails, as is shown here. They all have yeast infections. They have some other problems as well. And so you do get this broad whole system autoimmunity if central tolerance is messed up. Yep, Nadia.
I just want to clarify, because I'm not sure, why is this classified as autoimmune as opposed to immunodeficiency? We don't have that.
So this is classified as autoimmunity because the immune system is attacking the thyroid. The immune system is attacking the adrenal. The immune system should not be attacking those normally, but you have excess immune response. It's not like, so an immunodeficiency might be a patient who is missing T cells. That patient is not going to make T cell responses. Or a patient who's missing B cells. That patient is not going to make antibodies. That's immunodeficiency. Here, and we have autoimmunity, but you're right, it comes from a deficiency in air. But the end result is that you have excess immune response. The immune system is killing these self tissues that it shouldn't have. One other very famous defect in central tolerance that leads to systemic autoimmunity is listed here. This is a disease known as IPEX. And IPEX, as it shows here, stands for immune dysregulation, polyendocrinopathy, enteropathy, X-linked syndrome. It's X-linked, and what you might guess with something that is X-linked is that it is largely going to be found in males because they only have the one X chromosome, so they don't have the second X chromosome that could have a good copy. So if there's a mutation in the gene of interest here, then we're going to have a problem in males, and so we do tend to see this disease particularly in boys. Some of the issues that these boys have are autoimmune enteropathy, so autoimmune issues in their GI tract, eczema or other dermatitis, again, weird stuff with their fingernails, nail dystrophy, autoimmune endocrinopathies, so some type of attack on the endocrine gland, autoimmune skin conditions, could be alopecia, could be pemphigoid, could be a bunch of things. Huge secondary lymphoid organs, diabetes, food allergies, and infections. Most kids with this die within their first 2 years of life. And this disease is a mutation in FOXP3. And so if we have a mutation in FOXP3, we have an end to the idea of regulatory T cells. We don't have any regulatory T cells in this patient, and so we don't have any of the T cells that can turn off the other types of immune responses. And what you'll notice again is that this is a systemic problem. These patients have multiple autoimmune issues all at once because of this defect in central tolerance, as opposed to a more specific defect that usually occurs with peripheral tolerance. So at this point, we can think a little bit about autoimmune disease, and we're going to see this table twice. Here are a list of some very common autoimmune diseases, and in the column on the right-hand side, you can see the prevalence of those autoimmune diseases. So you can see that 1 in 50 people have psoriasis. You can see 1 in 100 people have rheumatoid arthritis, for example. And you might look at these and think, whoa, 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 whoa. You just told us all the reasons why central tolerance can mess up. Why is this not 100%? Why are there so few people with autoimmune diseases if central tolerance has all those flaws? And the answer is, hooray, we also have peripheral tolerance. So we can have all sorts of ways that our self-reactive cells are dealt with in developmental organs, the bone marrow for B cells, the thymus for T cells, but there are also ways that we can deal with those self-reactive cells in the periphery through mechanisms that are known as peripheral tolerance. And so, fortunately, yeah, central tolerance has a bunch of holes, but fortunately we have this other layer that's helping us out of peripheral tolerance. When I learned when I started really thinking about peripheral tolerance in terms of teaching peripheral tolerance, I learned 6 mechanisms of peripheral tolerance. And so I am going to talk through this process with the 6 mechanisms of peripheral tolerance today. So we will see these 6. It is possible that I will not have time to go through all 6 today. If not, that's what Wednesday's for. With each of them, I want to talk a little bit about how we make them happen, but also how they could be broken. How could that mechanism of tolerance get messed up to potentially lead someone to an autoimmune disease? And so we're going to see that for each of the 6. I will also tell you that I have an order of these in my head that I like, and the order that's in the slides is a different one, because I think this makes more sense for teaching, but I might be like a little off, because I'm like, this is not the right order for those, what am I doing? So if you were to think about, if you're going to try to guess how peripheral tolerance might work, based on what you learned previously about central tolerance, what might you guess would be a way that peripheral tolerance could work? You can notice central tolerance, and you could think about like common things. Yeah?
Does it stop like the clonal expansion of like cells?
Okay, so we can think about ways that we can stop clonal expansion of cells, and in fact, you're going to see that something related to that in a few of these mechanisms. But if you were just going to make a parallel to what you see with central tolerance, what might be the first thing you'd think of? Yeah, Cal. And so what could one of the things be that it leads to? What else? Death. We're going to get to anergy too. But the first thing is, well, we saw central deletion. We saw deletion as a mechanism for central tolerance. What about peripheral tolerance? Could we just have deletion again? And the answer is yes. The face I was making earlier was supposed to be my dead face. So the first mechanism that we see of of peripheral tolerance is called clonal deletion. And so it's possible that we can have some cells that may bind to self-antigen, particularly depending on the strength of that binding, and lead to deletion. There's a lot that's not really well understood about how this might work. You maybe could imagine it being related to exhaustion, whereas once the cell goes past exhaustion, that it goes to death and is actually deleted. So clonal deletion is one, it's relatively easy to understand in terms of what happens. How it happens, we got some questions. But what happens? Easy. Is it possible to break clonal deletion? So let's imagine that clonal deletion happened for some autoreactive cell in your body. Is it possible to break that deletion? What do you think, Marina? Why don't you think so? Yeah, because once the cell is dead, the cell is not going to become undead. So deletion is a little bit unique because once the cell has died, you're not going to bring it back to life. So once you did deletion, you can't break deletion. So unlike some of the other ones, we don't have a great way to break this one. And the reason why I've done the order that I have in terms of the one that I'm going to present second to you is it also is one that you really can't break. And so I'm ordering them in this way. So deletion is one of them. The next one I want to tell you about is based on some important information that we have learned recently, which is that B cell responses require T cell help. And so for a B cell to proliferate, to make high levels of antibodies to differentiate into a plasma cell, that B cell requires help. And so a very common mechanism of peripheral tolerance, and this one is especially famous for B cells. So like, yeah, you can think of ways it works for other cells, but usually when people talk about this mechanism, they're talking about B cells. Is a mechanism known as clonal helplessness. Your B cells all need T cell help, but your B cells are not tested against many self-antigens that are not present in the bone marrow. Fortunately, the T cells get tested against those antigens, and we delete the T cells that respond to those antigens. So those B cells have no partner to help them. The T cells all got deleted in the thymus, so the B cell might go into the periphery. It might require T cell help. It will, present its antigen on class 2, be ready to go, be looking for that CD4 T, CD4 T ligand interaction, and it'll get nothing. That B cell will be helpless, and that B cell will either be energized or at the very least will not get activated. And so because we have such a nice system of doing T cell central tolerance, that actually helps us out with B cells, because we don't have the T cells around to help the B cells. And so that allows us to have helplessness work well as a mechanism. What you will notice on this one is it's kind of something you already learned. It's just applying something that you've already learned in a different way. And for almost all of the mechanisms that we're going to see, that's true. And so I have definitely had students who've looked at stuff like this and been like, did we learn anything new today? And I'm like, yeah, but also no. So I can see, so you should be able to see how these things are related to things we've talked about before. So another one that we've talked about before, before that, can you break helplessness? No, because you can't make the T cell come back to life to help the B cell. So again, this one's not really breakable. I'm going to talk about the next two kind of together because it's hard to fully explain them separately. So the third mechanism of peripheral tolerance is clonal suppression. Apparently here we're allowed to use the word suppression. Nowhere else. And clonal suppression is just a fancy word for Tregs do their jobs. So in clonal suppression, we have our regulatory T cells with FOXP3, that transcription factor, and they are able to inhibit other cells and suppress the activation of those cells. And so because we have these Tregs that are able to do their job, we are able to, in fact, lead to some pretty good clonal suppression. There are some techniques out there that can help us Induce suppression. We know a little bit about inducing suppression as well. So there is this phenomenon known as oral tolerance. So what has been observed is that if one first encounters an antigen by injection, say, that one makes sort of a typical primary response and then has a major response later. However, if one first encounters an antigen by the oral route, the first time you see the antigen is because you ingested it, then in fact you seem to make more of a Treg response. And so here you can see some mice that were fed ovalbumin, which is our antigen, who then got ovalbumin injected. If the mice were fed control and then we injected ova, they make a huge ova response. But if we fed them first, the ova, and then injected them, they really don't make a response. So it's basically, it seems as though antigens you encounter through the GI tract lead to a Treg type response. This was actually an early set of observations about Tregs. This is when Tregs were called Th3s. And this kind of makes sense. If you think about many of the things, I have no idea what that is. I have no idea what phone is ringing. Okay. If you think about it, the things that you were encountering, orally, like steak, you don't want to make responses to. And so the antigens that you encounter through your GI tract should be pushed towards a more tolerogenic state. So it does look like the GI tract has a particularly immunoregulatory environment. There are specific downregulations of inflammatory immune responses, especially to those that you ingest, and specific macrophages and dendritic cells that help to tolerize to this. And so there are even a number of different therapies where people are doing things like trying to introduce specific allergens or other things orally to try to help with allergy or immunodeficiency. And so they're a little bit controversial. There's a lot that's not known, so I'm going to show you something. You're going to have 4,000 questions, and most of them the answer is I don't know. But just to show you, this was in Science earlier. There is this idea of doing oral immunotherapy for kids who have really high levels of peanut allergies and actually being able to decrease their peanut allergy by inducing somewhat of a tolerogenic response. It's with a very low dose because you want to be doing it in a way that doesn't turn their allergies on. It seems to work really well for the people who have sort of the super huge debilitating allergies to make it so like they can be in a classroom with other kids who are eating peanuts. It isn't going to make them eat peanut butter every day, but it brings it down into like a safety level, but it's very controversial. There's a lot that's not clear about how these sorts of things work. And so you can see this was in Science, October 18th of this year. And so it's something that people are still thinking about. There is a theory out there. I don't know enough data on it to give you my feelings on this theory, but there is a theory out there that one of the reasons why there might be an increase in peanut allergies is because more lotions have peanut oil on them, and so most babies are not getting their first introduction to peanuts orally. Most babies are now getting their first introduction to peanuts through their skin, through peanut oil in lotions. And so that's something that people are talking about. And so, the recommendation now is actually to give your baby peanuts as early as possible to try to get rid of peanut allergies. So clonal suppression is one of the big mechanisms of tolerance. And clonal suppression is related to the control of CD4 positive helper T cell subsets. So we saw some information like this. We would have seen information like this before if I knew how to go the right direction on slides. Now we have CD4 helper cells. CD4 helper subsets. And we saw that naive T cells can be biased towards the different CD4 helper subsets based on the cytokines to which they were exposed. And that will help push these cells into different directions. So that'll give us our different types of subsets. Hi. Okay. This is related to the fourth mechanism of suppression, which is a mechanism called clonal deviation. The idea with clonal deviation is that in any particular circumstance, the circumstance that is shown on this slide is an infection with a helminth, but we can think of a lot of potential circumstances. Some types of T cell, CD4 T cell subset responses are going to be good and protective, and other types of CD4 helper responses are going to be bad and not protective. So here we can see that if there was a Th2 response, we can have a number of different effects happen, all of which are protective when we get that helminth infection. But if we have a Th2 response or Th1 response occur to that same pathogen, instead we're going to have a damaging response to the host. And so the mechanism of tolerance here is that the immune system is producing the cytokines to push you towards a protective response and not towards a damaging response. So the immune response is deviating more towards a protective type response based on cytokines that are being produced, as opposed to a damaging type of response. The reason why I talk about deviation and suppression together is because deviation and suppression can both be broken in the same way. Both deviation or suppression can be broken by having the wrong cytokines provided to our helper T cells to push them to becoming a bad pathologic type of cell. So if we have the wrong cytokines made, we could push our cell from being a Treg and doing suppression into some type of helper T cell type that's pathologic. Or we could just go from the protective type, that, like the Th2 in the previous example, towards a Th1 that's pathologic. So in either case, the production of those specific cytokines is a problem. Deviation is usually the type of peripheral tolerance that students have the hardest time understanding. And so I want to tell you a little bit about some stories that maybe will help you understand deviation. So this is about Crohn's disease. We can make arguments about Crohn's disease in terms of how much it's autoimmune and how much it's allergic, and that's a topic for another conversation, but whatever. With Crohn's disease, we have situations where our intestine is actually having too much inflammation. We see damage to the intestinal wall, leading to GI tract symptoms. And what we can see is that Crohn's disease in this model only happens in one very specific situation. So we have to have mice who have some type of genetic susceptibility. The mouse has to have this specific gene. And if that mouse has a specific gene, it might be biased towards making, say, a Th1 response instead of a Th2 response. So the changes in genetics could change which subset the mouse is most likely to make. If that mouse happens to also get infected with a specific strain of a virus, so here you can imagine the red strain instead of the blue strain, or CR6 instead of CW3, the mouse will end up with Crohn's disease. A wild-type mouse infected with this will be just fine, but if we deviate the response that this mouse makes to this virus, suddenly the mouse is going to be making a pathologic response. Here it makes a perfectly fine protective response. Here it makes a perfectly fine protective response. But if we have this mutation that changes the subsets of CD4s a bit, suddenly this mouse is going to make a problematic response. And so this is sort of an example of deviation not working. One of the most famous of these is this paper that has to do with celiac disease. Again, we can have a whole argument at some point about whether celiac is allergic or autoimmune, or what exactly where celiac is classified, because that's a whole story in and of itself as well. But in any case, there's this really nice study on celiac disease. And what was shown is that most of the time, people have dietary antigens that they eat, shocking. Those interact with dendritic cells in their GI tract and lead to oral tolerance. So we have the dendritic cell leading to tolerance. We have T cells interacting with that dendritic cell. We have oral tolerance to those antigens. However, if a person is very unlucky and they got infected with a specific virus called reovirus, at the same time they first encounter this antigen, now they're going to be making this reovirus T cell response. They're going to be making Th1 cells responding to reovirus, and those Th1 cells are going to be throwing their interferon gamma all over the place in the GI tract. And now this T cell that normally would have been activated in a tolerogenic way, it would have gotten cytokines that were pushing it towards being a Treg, for example. This T cell is going to get a different set of cytokines. And now that cell is going to get pushed into becoming a Th1 cell instead of a Treg. And so now we're going to make Th1 cells against the dietary antigen instead of making Tregs against the dietary antigen. And we're suddenly going to make an immune response against this dietary antigen where we would not have before. And so one thing you can see here is that this is kind of a situation of bad luck. That you happen to be making that T cell response to that dietary antigen at the same time you had that viral infection. And so this is thought to potentially be a mechanism of celiac. This is just the same mechanism shown again. So normally dietary gluten comes in. It leads to a regulatory response. But if dietary gluten is first encountered at the same time as reovirus infection, then our cells are going to start making Th1 polarizing cytokines, and we're going to lead to a Th1 response and eventually the destruction of the GI tract. Whereas if this reovirus hadn't been around, this CD4 cell would have become a Treg and would have lived happily ever after. Yep, Molly.
I might be misunderstanding.
Yes.
Does that have anything to do with a failure in directional secretion as well?
That's an interesting question. Unclear is the easiest answer to that. Probably not, but I think that's a longer conversation that involves a few things that we haven't talked about yet. So, great question. Answer is a little beyond the scope. And any of you who went to the American Society of Virology conference this summer, you heard a talk from these people about this model. This is Terence Dermody, who some of you heard a talk from. So these are 4 of the mechanisms of peripheral tolerance. The 5th one is also one that you have learned about previously. And so if you think back to things we have learned in preparation for exam 2, is there anything else that you remember learning that has anything to do with peripheral tolerance? Any processes that seem related here to turning off a self-reactive cell in the periphery? Yeah, Malik?
Like the signal 1, signal
so we learned about the fact that T cells need two signals to be activated. There's signal 1 and there's signal 2. In fact, you can see that on this slide. So if a T cell receives signals through signal 1 and signal 2, that T cell is activated and proliferates. If that T cell gets signals through signal 1 and not signal 2, that T cell becomes anergic, and that cell cannot be turned on again later. And so our fifth mechanism of tolerance is clonal anergy. Clonal anergy is relatively easy to understand. We also understand a few ways that clonal anergy can be broken. And so clonal anergy has some specific pieces to breaking it. There's a fair amount we understand, and once you get this, you sort of get where a bunch of autoimmunity comes from. But in order to fully get this, I want you to think about something. So let's imagine we have a cell, an antigen-presenting cell, in fact, that is not infected with a pathogen. Does that antigen-presenting cell present any antigens? So it absolutely presents antigens all the time. It's presenting self-antigens. If it did not, then NK cells would be on the case, as we learned last time. So it's presenting self-antigens all the time. How many MHC molecules or MHCs, like, can you imagine would be on the surface of a cell? And this is, I know that I asked you questions like this on the exam, and I'm asking it in a slightly different way, so you might not get it. So let's imagine that we were going to really draw this antigen-presenting cell. And let's imagine that there's only one kind of MHC, not 3 class 1s and 3 class 2s. Let's just imagine there's one. So there's a class 1. Has a peptide. Is my drawing accurate? Or is there anything that is wrong with it? What do you think, Alina? How?
The ones that like present the cell.
Yeah. Any particular antigen-presenting cell does not have one MHC molecule on its surface. It has lots of them on its surface. Even, we talk about how there's 6 class 1s. You probably have many copies of all 6 of those class 1s. The surface of your cell is actually, learns how to draw, is completely coated with these. And then I got tired of drawing. But you can imagine going all the way around. Do you think they all present the same peptide? No, they're not going to all present the same peptide. And they're going to present the peptide sort of based on like the frequency of that peptide in the cell. So if there's some peptide that's present like 90% of the peptides in the cell, then it'll be on 90% of these. There'll be some kind of relationship like that. So here is the first mechanism of breaking anergy that we know of. Normally, an antigen-presenting cell at rest will be presenting self-antigen. It will interact with that T cell through signal 1, but there will be no signal 2, and that T cell will become tolerized. Specifically, that T cell will become anergized. However, if that antigen-presenting cell gets infected, the antigen-presenting cell is going to start making B7 to co-stimulate a T cell. That antigen-presenting cell is not going to have just one MHC molecule on its surface full of peptide. It's going to have many MHC molecules on its surface full of peptide. And so you can see that some of those MHC molecules are going to be full of the pathogen peptide. See, it's brown, just like the microbe, this little brown peptide. But it's entirely possible that there are going to be some MHC molecules on the surface that are still presenting self-peptides, because self-peptides don't go away. The cell doesn't stop making self-peptides. It's still making them at some rate. It still has self-proteins, and so there are still some self-peptides on some MHCs. You can see up here the self-antigen was yellow, and it's still yellow there. You can also remember that the B7 protein is a separate protein from MHC. It diffuses separately in the cell's cell bilayer, in the cell's cell membrane. And so it's possible that this B7 can pair with the MHC presenting the self-peptide, because this antigen presenting cell is just making lots of B7. It doesn't know which MHCs the B7 gets to go next to. It just makes a bunch of them. And we could turn on a T cell, giving that T cell signal 1 and signal 2 because our antigen presenting cell was infected. Thus turning on our T cell and letting it go attack tissue, leading to autoimmunity, because once our cell is activated, it does not any further require signal 2. So if the first time that T cell happens to meet antigen happens to be in an infected antigen presenting cell, then bad luck for you. You are going to activate that T cell. So does that make sense? Okay, so that's one way that we can break anergy. The other way that we can break anergy is through a process known as molecular mimicry. And so here you can see an example of molecular mimicry. You can imagine that one MHC molecule might present a couple different peptides. Remember that the T cell does not see the whole combination of the entire peptide. It just sees a few of the amino acids that are projecting up towards the T cell, and parts are projecting down. Well, it's possible that sort of the top could look the same between a pathogen peptide and a self-peptide. You can see that they're actually different. The self-peptide is white here, the pathogen peptide is red here. But the top part, the surface that the T cell sees, could look very similar. Thus, we could turn on a T cell in response to this pathogen peptide through correct signal 1, signal 2 interactions. Yet when this T cell leaves and goes out into the rest of the body, it might interact with an MHC molecule presenting the self-peptide that is so similar to the pathogen peptide. It's a molecular mimic. And suddenly we can see our T cell reacting against this self-antigen instead of reacting against this foreign antigen. And so this process of molecular mimicry is the other big way that we can sort of get around that perfect ideal of anergy. This is basically related to the same idea as cross-reactivity. So we can imagine that perhaps as a child, someone has a respiratory infection in the upper respiratory tract. We make some T cells, and it just so happens that those T cells bind to an antigen that's also found on beta cells in the pancreas. If we happen to have the same antigen from this respiratory virus and on the beta cells, the T cells that were activated against the respiratory virus, that got rid of the respiratory virus, would now make their way to the pancreas and would be like, haha, I found my antigen, and destroy those beta cells, thus potentially leading to type 1 diabetes. And so we can see this type of mechanism leading to potential problems. A lot of this has to do with, may have to do with ways that the T cell receptor and the antigen presenting cell can bind. So if you look on the left-hand side, in the blue, you can see the MHC. The green little skinny line is the peptide. And the red and the yellow are the alpha and beta chains of the TCR. And so you can see in the case of an influenza-specific TCR, there's a very specific angle of binding, and this has been shown in cartoon form on a few of the T cell slides that I've shown you. Sometimes The T cell is going to bind to the antigen presenting cell at a wonky angle. And in fact, sometimes you can get reactivity that's a little different than what you usually see, possibly leading to self-reactivity. So these are two examples of self-reactive T cell receptors binding to a self-antigen. In both cases, these are myelin antigens that are being presented, and these are T cells from multiple sclerosis patients, where you can see that the T cell is, just by binding at a slightly different angle, is suddenly able to recognize self-antigen. I have shown you these self-reactivity issues in the case of T cells, but it's also very possible for molecular mimicry to happen with antibodies and B cells. There is one very, very, very famous clinical condition where this happens. It involves making an antibody response to a molecule that's part of the streptococcal cell wall. And so here you can see a person potentially being infected with streptococcus that makes antibodies against streptococcus. It turns out that one of those antigens is actually cross-reacts in some people with a heart antigen. And so some people who have a streptococcus infection will end up with a condition known as rheumatic fever, which is an autoimmune destruction of the heart by antibodies that were cross-reacting to streptococcus. And so molecular mimicry is not just a T cell thing, it's also a antibody thing. What I think you might realize at this point is the potentially important role of infection in autoimmunity. And so what you've seen are a number of different ways where there's sort of bad luck plus infection that led to the breaking of tolerance with these mechanisms that we've seen. And epidemiologically, there are a number of different links that people have seen. Usually, there has to be a case of a specific HLA type that presents that mistakenly cross-reactive antigen. So you have to have the bad luck to have that HLA type and that infection and that weird timing of that infection to get that autoimmune disease. This also can be somewhat difficult because oftentimes that infection happens long before the autoimmunity presents, and so we sort of have to go back in time and try to guess what infections you had, and that is not always very easy to do. But you can see that there is, for example, some relationships between type 1 diabetes and things like Coxsackie virus, echovirus, or rubella. You can see there are a number of bacterial infections that seem to be related to arthritis, and there are far more than all of that. And so these are sort of the really big mechanisms of tolerance. I want to start talking about the sixth mechanism, though I'm not going to be able to get all the way through the sixth mechanism. The sixth mechanism is the biggie, or not the biggie, but it's the one that is the showstopper, I guess, one that everybody gets excited about. So the sixth mechanism is one that doesn't follow as obviously from the things that you've already read, whatever. And it is known as clonal ignorance. So you'll see that they all have clonal in their name. It's usually called clonal ignorance when we want to make it all matchy-matchy with the other ones, where they all have some name, clonal something or other. And so we can call this clonal ignorance. But it's also often referred to as immune privilege. And so I typically talk about it as immune privilege. There are certain parts of your body that are known as immune privileged sites. And some of the big immune privileged sites are listed on this slide. If you look at this list of anatomic locations, what do you notice? Or what do all of these things have in common? Yeah, Cal. Yes, but we haven't gotten there yet. So before that, what do they have in common? Would you, evolution-wise, not even evolution-wise, would you be very good at surviving and reproducing if any of those got destroyed? No. These are super important things that kind of have to be kept safe. These are things that evolutionarily we can't go without. We cannot survive and reproduce without these things. We lose these, we're in some bad shape. And as a result, the classic definition, which is exactly what Cal just said, is that you don't have immune responses in these locations. We're going to sort of play with that a little bit and think about whether or not that's true and exactly what's going on. We're not going to have time to go through all of that today. But I'm going to close off with the classic example of immune privilege. I'm going to tell you about this immune privilege disease. Everyone freaks out when they hear about it. And then we're going to talk more about the specific details of immune privilege, how it may or may not work. But the original definition of it was largely based on this particular disease called sympathetic ophthalmia. And I will tell you that 2 years ago, I think it was 2 years ago, once upon a time, I taught sympathetic ophthalmia while a student in my class had just had an eye injury, and I didn't know what to do, because it's about eyes. So the eye is one of our immune privilege sites. And it has been observed that if one has trauma to one eye and has damage to their eye, that can in fact lead to eye antigens leaking out of the eye and going to the draining lymphoid organ. And we realize, and what is seen to happen in these patients is that we activate T cells in response to those eye antigens, and those T cells go back and destroy not just the damaged eye, but also the non-damaged eye, leading to destruction of both. And the idea has always been that at the beginning, the antigens in the eye were sequestered. They were basically hidden from the immune system, so that we didn't ever make responses. The eye was just too important. We didn't want any responses to happen there, so we basically kept the immune system out. We sequestered that area from the immune system. Yet, if the antigens came out and we turned on the T cells are like, woohoo, I found a new antigen, and will then attack all places where they see this antigen, this new novel antigen that they've never seen before, and in fact destroy both eyes. And the reason why it's important to point this out is that this is sort of the classic idea of what immune privilege always meant. Immune privilege in the past has always meant you keep the immune system physically out. There is a physical barrier to allowing the immune response in. The slides that I haven't finished today that I'm going to start with on Wednesday are going to sort of give some other evidence and help us understand what we currently think is going on with immune privilege, because it's a little more complicated than just you build a wall. There's a bit more going on at this point. Marina, did you have a question?
Yeah, for the eye, as it being an antigen, I thought the thymus took care of it.
So it seems as though some of these antigens are not negatively selected. Yep, Alina.
So by trauma, is that trauma and like an infection?
No, usually it's like getting stabbed in the eyeball.
Oh.
It's you break the back of the eye, so the antigens are coming out. So we will finish talking about immune privilege and then talk about some other important biology of autoimmune diseases on Wednesday. I will see you guys then.